We move now to the third presentation from Manoel Oliveira de Moraes, Jr. from University of Sao Paulo in Brazil, School of Pharmaceutical Science, Department of Clinical and Toxicological Analysis. And you're going to speak about melanoma cells and, I think, treatments with copper.
Hi everyone, good morning. Thanks for having me here. As an introduction, I'm Manoel from the University of Sao Paulo in Brazil, and today I will present the results of my PhD project, Antiproliferation, Antivision, and tyrosinase-like activity of new copper complex in melanoma cells. So out of context, melanoma is the very important problem in human health. So it's the worst type of skin cancer with the highest rates of death. It has its name because of it comes from the melanocyte, which is the cell that produces melanin in the dermis. It has this worse prognosis compared to other types of cancers, mainly related to the hyperactivation of the MAP kinase pathway that controls cell growth, invasion, survival, and problems with the inhibition of apoptosis. And the standard of care of treatment comes with the inhibition of the BRAF, which is the most type of mutated protein, is the MAP kinase pathway, and also as a single or associated with the MEK inhibitor. However, as a therapeutic challenge of most target therapy, it started the appearance of chemo-resistant phenotype and therapeutic relapse within the time, usually after 6 or 12 months of treatment. So the patient start to have in the growing of the tumors and the metastasis again. And this happens mainly because of the intratumoral heterogeneity that confers the ability of these cells to trigger different types of mechanisms, such as the hyperactivation of compensatory pathways, upregulation of the growth factors, metabolic reprogramming, and so on. And that's why it's so important to try to find new pharmacological treatment alternative against melanoma, especially melanoma with resistance phenotype that could overcome these resistant mechanisms, and also with better tolerance and less adverse effects. In this context, we have metallodrugs, which are drugs with metal on this structure. One of the examples is the copper complex that could have been an advantage of having a conformational versatility comparing to only organic compounds, and also because of its metabolic type of metal that could be more tolerant in terms of treatment. We see an increasing of the importance and publications over time with copper complex in general, and also with anti-cancer activity. And the copper is very important because it has this physiological role in many types of cells, including melanocytes, as it controls also the activation of the MAP kinase pathway, being a cofactor for the activation of MAC, and also controls the activation of OAK2 that controls autophagy. So especially in tumorigenic cell lines and melanoma, it can be modulated. The level of copper can also increase by having a cytotoxic activity, or decrease having a cytostatic activity. Also, this MAP kinase pathway that is controlled by also copper can also modulate the activity of MITF, which ultimately controls proliferation. I'll show you later why. And also it controls the activation and the synthesis of tyrosinase, which is an enzyme that is very important in the formation of the melanin in the melanogenesis process. So we see also that copper is very important in all of this process in melanoma cells and melanocytes. In this context, we have the Institute of Chemistry in Brazil, the collaboration that this group synthesized and studied the biological activity of new copper complexes, being mono and dinuclear. And so we see here the structure of them, and they show in 2 different papers that these copper complexes can have cytotoxic activity against melanoma cells with double-stranded DNA cleavage, oxidative stress, and increasing of autophagy features, but also a tyrosinase-like activity. So in this context, we ask ourselves, does this compounds have activity against melanoma with BRAF resistance phenotype? So the aiming of this project was to investigate the cytotoxic activity and the mechanism of action of these mononuclear and dinuclear copper complexes against melanoma with cell lines with BRAF resistance phenotype. So as methods, we use different types of melanoma cell lines and fibroblasts with different mutation settings. We developed a resistant phenotype trying to mimic what happened in the patient by treating those cells over time with increasing concentration until a plateau. And we also see how the cells keep upregulating or keep activating the MAP kinase pathway, even though in the presence of vemurafenib, which is a BRAF inhibitor. And also we compare with cisplatin, which is a metallo drug very important in the treatment of cancer, and dacarbazine, which is a standard of care to treat melanoma. As methods, we use classical viability assays such as MTT, triple blue exclusion, Western blot to understand the cell signaling, and also flow cytometry, other types of techniques to understand how this copper complex goes. As a first result, we show that the dinuclear copper complex is a cytotoxic against melanomas. We see here a table of IC50. In the first row, we see cisplatin, dacarbazine, the mononuclear was not cytotoxic against these melanoma cells, even though they used to treat in the patients with over 100 micromolar of IC50. But comparing to the dinuclear copper complex, it has a IC50 in the low micromolar setting in both parental and resistant cell line. And with a more strict approach with triple blend with only dinuclear copper complex, we see the same pattern of activity in parental and resistant cell lines all across the board. And that's why we choose the SCAML28, parental and resistant, they had the same potency between both. So we're going to play with 15 micromolar as a concentration to understand the mechanism of action of this copper complex. So first of all, we show here that this complex trigger autophagy signaling by increasing the lipidation of LC3B in its active state in both parental and resistant cell line, but also in a resistant cell line, we see a downregulation of P62, showing that not only the formation of autophagosomes is happening here, but also the degradation of the cargo too, showing that this copper complex causes triggering of autophagy signaling. In a apoptosis setting, the cell death part, we use flow cytometry to assess the phosphatidylserine acetylation. So we're showing here a graph of cell population that after using 30 micromolar, we see a decreasing of viability and increase of late apoptosis morphology in both parental and resistant cell line. And when we treat those cells, we pre-treat those cells with ZVAD, which is a pan-caspase inhibitor, we see the same setting, the same pattern as the cell keep dying by 30 micromolar of the dinuclear copper complex. And also we see a downregulation of BCL2, which is a protein related to apoptosis or blocking of apoptosis, showing that this might be one of the mechanisms of cell death. In terms of cell cycle modulation, we use PI to see aided propedium to see the quantity of DNA and try to understand the cell cycle arrest, and we see here the nuclear corporate complex triggers the cell cycle arrest in the sub-G1 phase, which means that we have more cell lines, more cell population with cells with less DNA content than the healthy cells, showing that we're having a DNA damage caused by the activity of the dinuclear corporate complex. And also, a difference between the parental cell line being less sensitive comparing to the resistant cell line that has more DNA damage compared to the parental, and also the cleavage of PARP, which is a factor of apoptosis, but also shows the DNA damage signaling, and it shows here that the resistant cell line is less active in terms of PARP activity in general, so it may explain why the resistant cell line could be more sensitive to DNA damage. In terms of mitochondria activity, we're showing here basically a less uptake of tetramethyl-rhodamine, which is a stain that gets clogged in with the healthy mitochondria. So both parental resistant cell lines gets less of tetramethyl-rhodamine in this mitochondria, showing that also the dinuclear corporate complex can cause mitochondrial membrane disruption in both parental and resistant cell line. In terms of oxidative stress, we treat those cells with 30 minutes of incubation, and then we follow the kinetics of 3 hours of activation of ROS, inducing of ROS. So in 0 minute, we barely see a difference between the groups. But when we treat those, when we see after 3 hours, even the concentration, they are not cytotoxic like 7.5, we see increasing of oxygen species, reactivity, and all of the cell lines, both parental and resistant cell lines, and also it could be downregulated and blocked by the using of antioxidant agent as the N-acetylcysteine. In terms of gene modulation, we, as with the collaboration of Professor João Machado Neto, we see a panel of genes related to cell cycle, apoptosis, and autophagy. And here, we see that in parental cell lines, a downregulation of genes related to the blockage of cell cycle in blue, and also a upregulation of genes related to apoptosis and the blockage of the cell cycle, showing that this compound can block cell cycle in general and prevent proliferation. In the same pattern, but less extent, we see the same thing in the resistant cell line, which is kind of what we expected, since this cell line is more resistant to trigger apoptosis and to stop cell proliferation in general. In terms of morphology and migration, we see here that the nuclear corporate complex can cause antiproliferation, anti-invasion activity, as we see the migration of the blockage of closing the gap in the wound healing assay after 48 hours, that the vehicle can close almost 100% of the gap, but 70.5 of micromolar of the nuclear corporate complex block all the migration of the cells. Also, in terms of invasion, we use double bonding chamber with matrigel between both compartments, and we show that 7.5 and also 15%, 50 micromolar can block the migration of the cells to the different compartment through the matrix, extracellular matrix mimetic. And also a downregulation in the resistant cell line of AXL, which is a marker of invasion, very important in the phenotype of these cells. Also, in terms of colony formation, we treat those cells for 2 weeks and treat them for twice a week, and we see low concentration that are not causing cell death, blocking of proliferation in 3 micromolar and 7.5 micromolar in both cell lines. To a more complex model, I think we're familiar with this model here. So it's a reconstructed human skin. So we make the dermal equivalent with collagen fibroblasts, and then we put keratinocytes, melanocytes, and melanoma to construct the whole skin, and treat those cells with the dinuclear copper complex, and then stain with H&E at the end. We have different types of cells, both naive and resistant. I'll try to guide you here. So we have first the not treated cells without the melanoma, and treated cells without the melanoma with 30 micromolar, treated 3 times with 3 cycles. We see a barely difference between the skin without the treatment and the treatment, showing that the dinuclear copper complex is not really toxic against the skin itself. But when we put the parental cell line, we see a lot of cells in the dermis being accumulated and being proliferated. And when we treat those cells with 60 micromolar and 30 micromolar, 3 cycles, we see less cells being stuck in the dermis comparing to the control. Also, when we treat those with the resistant cell line, we see not treated, a lot of cells being invading the dermis, as we see, as a part of its phenotype switching. And when we treat those cells with 60 micromolar and 30 micromolar, we see with 30 micromolar a complete depletion of the cell invasion and the melanoma invasion through the dermis. As we already mentioned, the phenotype switching between those cells shows a more MITF happening in the parental cell line with a more proliferative and pigmented type of phenotype. And also the resistant cell line, which more AXL, less MITF, but more invasive, more mesenchymal. So in practical, we see here the peak of these skins. So as we see no treatment, first of all, comparing the parental with the resistant, we see no pigmentation with the resistant cell line, but we see a pigmentation of the parental skins with the more, as we use more cycles of dinuclear copper complex. So we decided to try to understand what happens with the skin in terms of pigmentation here. So we did the skin again, we reconstructed the skin, but this time without melanoma, only with the melanocytes, and we used the pigmented skin with a supplementation of tyrosinase to try to mimic what happens in the body. So here we see first the skin in the top, the skin's here, the non-treated, a little bit pigmented, as is supplemented with tyrosinase, with more melanocytes. But also, when we compare with the treatment with the dinuclear copper complex, 15 micromolar, which is not really that's toxic against the skin with one treated. But when we treat with 3 cycles, we see increasing of pigmentation comparing to the 30 micromolar 2 cycles. And when we see these in the Fontana-Masson stain, we see the cells here non-treated with no melanosomes here. But when we compare the regular media without the tyrosinase, already the dinuclear copper complex causes an increasing of melanosomes, as we can see here in the blue arrows. But comparing to the supplemented media, supplemented skin with the tyrosinase, we see also already melanosomes in the skin not treated, but when we treat with the dinuclear copper complex in both 30 micromolar and 15 micromolar, we see increasing of melanosomes in general, showing the induction of melanogenesis and in this complex 3D model. So as a take-home message, we show here that this copper complex is cytotoxic against melanoma cells, triggering autophagy signaling, inducing cell death, feature caspase independently, causing DNA damage, leading to mitochondrial disruption, produce oxidative stress, and also was anti-proliferation and invasion in both monolayer and skin cell model, and also ultimately it has a tyrosinase-like activity, increasing the melanogenesis in 3D skin model, showing that this copper complex had not only importance in trying to understand the activity of importance of copper in homeostasis of the skin, but also this copper complex can have a dual function, suggesting a more broad application in dermatological and oncological therapy. So I'd like to thank the SPIM committee, the audience, and also our collaborators and our group in Brazil. Thank you.
Thank you, Manuel. Yes, question, Federico.
Thank you for your very complete presentation. In the beginning of your talk, you show a slide about the treatment of melanoma, and you spoke about BRAF inhibitors, MEK inhibitors, but you haven't spoken about PD-1, PD-L1 inhibitor. You spoke about the cells, but not the immunity. And right now, the main treatment of melanoma, metastatic melanoma, is PD-1 or PD-L1 inhibitors. So my first question is how can you put in perspective your dinuclear copper component in the field of treatment of melanoma?
Yeah, it's a very important question. We don't, in terms of like the skin, we treat those cells with the skin. We are developing a more broad model of the melanoma with more immune cells, so we can maybe test this copper complex in the skin with more cell lines with immune system, as we think we could see a difference between this copper complex with more complex model in the skin, but we don't have really a connection between the immunomodulators and the copper complex yet.
Okay, and another comment is that do you think that because the BRAF inhibitor were very important, and it was really a very important improvement in the management of patients with metastatic melanoma. But unfortunately, you have resistance. So can you imagine that it will be different for your dinuclear copper compound?
Yeah, we have a perspective of trying to combine the vemurafenib and the MEK inhibitor with the copper complex with lower concentrations to see we can delay the possible resistance phenotype arising or even resensitizing the cells of the inhibition of BRAF.
Okay, and about the induction of skin pigmentation, do you know human disease? Is it a surprise for you, or do you think it was expected?
It was expected because the previous work that synthesized those complexes showed that this complex could catalyze the conversion of tyrosine in L-DOPA. So it was something that we expected, but it's always a surprise when we see the skin, because we're trying to see the inhibition of the invasion, the proliferation, but when we see the skin, it was something that okay, this is a very interesting topic, we need to search a little bit more. So we have a little bit of plans to try to understand more this mechanism and why it happens, because we know that it's a tyrosinase-like activity, but how we don't know exactly the mechanism yet.
Because in fact you have a human disease in which you have accumulation of copper and you have skin pigmentation. Do you know this disease?
I'm sorry, can you repeat?
there is a human disease in which you have accumulation of copper and at the same time skin pigmentation.
We are thinking about more diseases that has like hypopigmentation and like from post-inflammatory that causes hyperpigmentation. Some patients can be benefited by the increase in activity of tyrosinase that at the end could cause melanogenesis. As some works are showing that like more copper can treat those situations.
But you have Wilson disease in which you have accumulation of copper and you have pigmentation, skin pigmentation.
At the top of my head, I don't remember diseases that are related to pigmentation, and we know that more copper can cause hyperpigmentation, but I don't know. It maybe can be a side effect in these cases.
Thank you.
I have a question about the type of DNA damages that are generated by your treatment with the copper. Did you check this specifically? If there are double-strand breaks, oxidative modification of the DNA?
They cause double-strand break of the DNA.
Okay. You did some gamma-H2AX immunofluorescence or to
It was a immunofluorescence, but it was a work before my project. Yeah.
Other questions in the audience? I have also a question about the receptor, the copper receptor. Did you check the expression of this receptor on the cancer cells or melanoma cells compared to normal cells?
No, we didn't, but it's a perspective of trying to block the receptors and see if the cells can still have the uptake of this compound and see if the cell dies less or are more invasive compared to the control.
Okay. So if there is no question, thank you very much for your presentation. Thank you all for the discussion.